Hi, everybody. This is Dr. Nigro with a chem calculation screencast. The screencast is going to focus on the experimental technique of chromatography. Now, this is not a heavy calculation-based technique, but it is an important one when you're dealing with mixtures. So I wanted to make sure that everybody understood what we were talking about when it comes to chromatography. All right, so it is an experimental technique. And I don't mean experimental in that we don't understand how it works. I mean, it's one that we use when conducting experiments. And it is used to separate the components in a mixture. All right. So let's say you have conducted an experiment in the lab, you've produced some products, but it's a mixture of products, and you want to now separate those products into individual products. Chromatography might be one way to do that. All right, now the separation uh, is due to differences in some physical properties of your components. All right, when we talked about our classification of matter, remember we talked about what a mixture is. And a mixture can be homogeneous or heterogeneous. Now, when you do chromatography, it tends to be a homogeneous mixture. There are other techniques that work better for heterogeneous mixtures, but it's a mixture nonetheless, which means that the chemical entities are physically associated with each other, but not chemically associated with each other. All right. So the technique is not going to break any bonds. It's not going to form new bonds. There'll be no chemical change that occurs when you conduct a chromatography experiment. It's just going to be the separation of these components based on some difference in their physical properties. All right. Now, some examples of what you might exploit: size. Another great one is polarity. And then there can be some column materials that are very specific to some sort of binding interaction. All right. Great example of that. Let's say that you're conducting an experiment and you produce a series of antigens. All right. And those antigens are each very specific for what type of molecule they will interact with. So if you can build a chromatography setup that takes advantage of that, then you can pull out a particular component of that mixture. All right. Now, the experiment itself, there are two phases involved. All right. The first phase is called the mobile phase. All right. This is a solvent that flows and carries the mixture. All right. Now, it's important that you choose your mobile phase based on what your components are as well. Sometimes if you have a polar substance that you want to leave behind and a nonpolar that you want to separate out, maybe you use a nonpolar solvent to carry that nonpolar piece along with it, or vice versa. Maybe you want to pull the polar piece out, use a polar solvent. All right. But it's the solvent that moves through your setup and it carries the mixture components with it. All right. The second is your stationary phase. All right. Now, this does not flow, it does not move, and components, oh, I spelled that wrong. Interact with it. All right. So, what does that mean, interact with it? It can be a couple different things. They can be absorbed onto that system, they can move through that mobile phase. It could be that the mobile phase has some sort of chemical entity attached to it and that interacts with one of your components. It could be you have beads that have pores in them and smaller molecules get kind of stuck in those pores, moving through a slower path where larger molecules just bypass it altogether. There are a lot of different choices for this stationary phase, and it just depends on what you are trying to do, what you are trying to separate out. All right. So, what information can we get? Well, one of the biggest pieces of information that we can get is called the retention factor, which is sometimes abbreviated capital R, lowercase f. All right. And what this is a ratio to show how far a, oh, I'll let this back up there, a component moves with the mobile phase. All right. There are many common setups for chromatography experiments, which means that there are known retention factors for many components of mixtures. So if you set something up based on a similar experiment to where we have known retention factors, then you can use it in some ways to identify what components you might have. Now, retention factors can be very similar from component to component. So it's not the only thing that you can use to identify your components, but it is one of them. All right. How do you calculate that? All right. That retention factor is the distance of your component divided by the distance of your mobile phase. All right. So it's a very simple calculation. All right. So if we look at our chromatograph above, all right, this is our chromatograph right here. All right. Let's look at our, I don't know, I guess we'll go with red. Let's look at red. All right, first of all, you can see that the solvent front is all the way up here. So the solvent moved 10 centimeters during the experiment. The red component of our mixture moved 2 centimeters. All right, so the retention factor for that red would be 2 centimeters divided by 10 centimeters, which is going to be equal to zero point 0.2. So that would be the retention factor for the red component. All right, you can see the purple component would have a different retention factor as with the blue. All right, now the other piece of information that I can get from this chromatograph is the number of components within my mixture. I can clearly see that I have 3 components, red, purple, and blue. Okay, all right. Now that's paper chromatography, which is what we tend to do in AP Chem, right? But there is another technique that is very handy. I'm going to very briefly talk about it. I have a picture shown here. This is called column chromatography, all right? If you do any sort of organic chemistry or biochemistry, column chromatography is your friend, all right? In this case, instead of it having a flat two-dimensional piece of paper that we're allowing our particles to move through, we're going to have a column packed, whoops, with our stationary phased. All right. This material interacts with our mixture, slowing some components and allowing for separation. All right. If you're like me and you worked in a biochemistry lab for several years, I used to purify proteins. And as part of that purification process, I would need to separate multiple protein components from one another. And one of the easiest ways to do that was to separate them based on size. All right. Some proteins are very large, some proteins are small. So I had a very, very large column. All right. It was about 5 feet tall, had a diameter of probably of about 2 inches, 3 inches. So it was large. And it was packed with a stationary material comprised of porous beads. So particles that were smaller would take longer pathways because they actually could enter the pores and move through the beads, whereas larger particles, larger proteins could not. So they would just flow straight down the column. But in the end, it allowed me to separate these components based on their size. At the bottom, I had a test tube rack, and I would sit there, and as a particular band of proteins came off the bottom of the column, I would collect them in test tubes, and it allowed me to separate my proteins from one another using column chromatography. All right, so it allowed for separation and collection. Now, I always had to go do a bunch of other stuff with those to make sure it was the right stuff, but that was one of the many steps that I did as part of my protein purification. All right. Now, for us, it's going to be mostly paper chromatography, which is where we'll have a piece of filter paper. We'll put our samples on the bottom down here. All right. We'll set it into our mobile phase, and then we will allow the mobile phase to run up that piece of paper through capillary action. All right. Then we'll take some measurements. All right. So if we look at this particular setup and we look at our dots, how many components were in the mixture? Well, I see two, my green component and my red component. If the mobile phase were water, which sample was more polar, red or green? All right, so first thing you got to keep in mind is water is polar. So substances that are polar are going to move with the water, whereas substances that are nonpolar are not going to move as quickly with the water. All right, so I'm going to say then that green is also polar because moved more readily with that mobile phase. All right. And then calculate the retention factor for each. All right. So the retention factor, let's start with the green. All right. So if we look at our measurements, all right, it looks like the mobile phase moves 6.5 centimeters. So that's going to go on the bottom. And our green component moves 6 centimeters. So we're going to get a retention factor there of 0.92. All right. We then repeat this time for our red sample. Again, the mobile phase moves 6.5 centimeters, but it looks like our red component only moved 2 centimeters. So that gives us a retention factor of 3.1. All right. All right, so I hope that this introduction to chromatography has been useful. Remember, it is a technique used to separate components of a mixture based on some difference in a physical property. All right, that technique can be anything from paper chromatography to column chromatography. There's all types of chromatographic experiments that you can run.